Hi class, we're going to finish up chapter 9. In the last video, we were reviewing the basics of, well, it wasn't really basics, it was hardcore stuff. The neuromuscular junction, the events of the neuromuscular junction, and the basic anatomy of a muscle fiber. So now we're going to look closer at the sarcomere and its action while the contraction is going on, and the recovery and variations and so on and so forth. So, let me switch the mode here on my little computer. There we go. So we had just talked about how the excitation contraction coupling happens. So in other words, as soon as that sarcolemma is excited by that motor neuron, that excitation or action potential is going to travel down the T tubules, cause a release of calcium from the terminal cisterna of the sarcoplasmic reticulum, and that calcium release is going to trigger contraction, and the sarcomere shortens. So each myosin head is already energized and basically cocked and waiting to join up with the actin. So the energy is supplied by a breakdown of ATP by myosin, and the myosin head kind of acts like an ATP ACE. ATP ACE is the enzyme that creates and destroys ATP. So the breakdown products are ADP and P, right? Adenosine diphosphate and phosphate. They're still attached to the myosin head. So click there. There we go. So here you see our resting sarcomere, and what's happening is our myosin heads are cocked and ready with the ADP and that extra phosphate waiting to be activated, basically. These calcium ions are starting to come in, and now our contraction cycle begins. As soon as those calcium ions arrive at that zone of overlap, the active sites are exposed because calcium binds to troponin. And when calcium binds to troponin, it's going to weaken the bond on the actin-troponin complex, and troponin changes shape. It's going to move out of the way. And what that's going to do is reveal the active site that's on the tropomyosin. Tropomyosin has the active site for where the myosin head needs to bind. So as soon as troponin is out of the way, because calcium's there to sweep it away, myosin jumps up, touches that thin filament, and that cross bridge is formed. It's called a cross bridge. When I can probably do it. I don't know. It's awkward. So here's my myosin head, here's my actin, and as soon as that calcium comes in, removes the troponin, that myosin can bind and shift and move, not that dramatically, I can't move my arms like that, to actually move and shorten the sarcomere. So that's the head pivot. The myosin head will pivot. That's that kind of jerking motion that the myosin head does. And it does that with that stored energy. It's called a power stroke. So the stored energy, that ADP that head has, is going to be released, and then the cross bridges can detach, and then a new ATP can come and bind to the myosin, and then the myosin can use its ATPase to break down the ATP into ADP plus P, and it's kind of cocked, loaded, and waiting and ready for another cross bridge to form. So that active site can, once it's available again, when calcium binds again, that myosin's ready to jump on up there. So the myosin has to reactivate itself, and it uses the ATP to do that. It's basically splitting an ATP into ADP, recocking itself, getting recharged. And that is the final step of our excitation contraction coupling. It's the contraction cycle. It's what I just described. Your Mastering A&P website has a really good video on it. You can go into Google World or Internet World, type in animation of muscle contraction, animation of sarcomere, excitation contraction coupling animation, etc. There's 1,000 of them out there. Just make sure you're looking at it from a credible source and not like little Jamie in her basement making a video. Although obviously I know what I'm talking about. Not everyone does. So the muscle fiber contraction cycle is exactly what I just described to you. Every contraction cycle is going to continue to be repeated as long as calcium remains in that sarcoplasm. So the T tubules have to soak back up or not the T-tubules, the sarcoplasmic reticulum, have to soak back up that calcium. They have to have a calcium reuptake. So as soon as those calcium channels close, calcium channels or calcium pumps are going to pump calcium back into the terminal cisterna of the sarcoplasmic reticulum. The troponin tropomyosin complex moves back over the active sites, so they're no longer revealed or no longer naked, and the myosin cannot bind to the actin. So no cross bridges can form. So here's the whole thing. Our resting sarcomere, our calcium has just been released, our myosin heads are cocked and ready with ADP. Now calcium comes in and binds to the troponin, revealing the active site on our, there we go, revealing the active site on our tropomyosin. Now look, our myosin head has now binded or formed that cross bridge to our actin. And now the power stroke, those heads pivot, and look what happens, that ADP is now released. Our sarcomere shortens, that's why you see this little white arrow pointing this way. And now, in order to detach the head, ATP has to come in, and ATP is broken down by ATPase within the myosin head into ADP plus P. Our calcium's still there, our myosin's still recharged, but the very last step, that's showing you the contracted sarcomere, the very last step would be the reuptake of that calcium so that cross bridge cannot form again. You can pause the video here, go back and answer these 3 questions. Let's keep moving. Here are some functional properties of skeletal muscles. We're moving into another little section here, and I'm going to zip through these outcomes and get into the connection between excitation, contraction, and muscle tension. So excitation has something known as neural control. In fact, everything in our body has some type of neural control mechanism to it. Our skeletal muscle fibers are stimulated by a motor neuron at a junction called the neuromuscular junction, or NMJ, as we talked about in the last lecture. The stimulus is an action potential. That action potential is an electrochemical gradient that can be propagated or carried down the axon and into the muscle fiber. Excitation is when that action potential, AP, that's what this AP is abbreviated for, an action potential is going to cause the release of the acetylcholine into the synaptic cleft. The acetylcholine binds to the motor end plate, opens sodium channels there, leading that action potential into the sarcolemma, down into T-tubules, so on and so forth. So that is ultimately causing calcium ions to be released from the terminal cisterna. So when those calcium ions are released, that's when the contraction cycle begins. Calcium binds to troponin, exposes the myosin. Myosin can then form the cross bridge. And as long as ATP is available, ATP and calcium, that cycle is just going to keep going. That muscle is going to continue to contract over and over and over again. With that contraction, the sarcomeres are shortening. It doesn't seem like it's going that far because it's just this little tiny movement. But if you have thousands of these sarcomeres lined up against each other and they're all moving a little bit, that's going to produce a big motion. So the thick and thin filaments interact in that cross bridge formation, and it's ultimately producing tension on the muscle because the muscle is shortening, that's pulling on the tendons, and that's where that tension comes from. So here's the whole thing in a nice cool diagram. I would love for you to redraw this, paraphrase it, summarize it, or write each step on a note card. Mix up the note card, see if you can put it in order. It's a great way to study this complex process. But this is where all the magic happens. That action potential coming down the motor neuron, releasing acetylcholine from the chemical synapse here into the synaptic cleft, being released from those vesicles, binding to the motor end plate. Acetylcholinesterase is waiting in there to gobble up the extra acetylcholine. Sodium channels open. As those sodium channels open, that's going to cause excitation down the T-tubule. That's going to communicate to the terminal cisterna to release calcium. Calcium is going to bind to troponin. Troponin is going to move. The myosin heads can now bind to our, where are we? I think we're over here now. Our myosin heads can now bind to our actin, and our muscle fibers shorten with that power stroke. Boom. Here's the whole thing again in individual slides for your viewing pleasure. All right, you can pause the video, go back and review if you need to. Let's talk about how sarcomere length affects tension. There's an optimal length or an optimal distance that our thick and thin filaments need to overlap. In other words, Tension does not depend on the number of sarcomeres that are contracted. In fact, upon stimulation, those calcium ions that are released are going to go to all of the triads within the muscle fiber. So, in fact, all the sarcomeres in that fiber are stimulated. They all contract together. So they're basically either activated or they're not activated. The muscle fiber is either on or it's off. That motor neuron has either contacted that muscle fiber or it's contacted another muscle fiber, right? It's not half of the muscle fiber contracting and the other half not. So our sarcomeres basically have to have an optimal resting length. In other words, this is going to maximize the number of cross bridges that can form. The more cross bridges that can form, the more powerful the power stroke, the more tension that can be produced, the more movement that can be produced, et cetera, et cetera. So the normal range of sarcomere length is between 75 and 130% of its optimal length. So in other words, you want to get it right in the middle. So the tension at the stretched sarcomere lengths is basically going to affect whether or not well, it's kind of the other way around. How short the sarcomere is at rest is going to affect the amount of tension that can be produced. So in other words, if the sarcomere is already pretty short, if that zone of overlap is already pretty big, well, the sarcomere can't move much at all, right? And if the sarcomere is already pretty stretched out and pretty long, then the zone of overlap is very small. So there's not going to be much grabbing going on, many cross bridges forming. So again, affecting the tension that's produced. So there is a maximum or optimum length that sarcomeres need to be. This is just explaining the same situation that I just explained. So at optimal length, greatest tension can be produced by the maximum number of cross bridges forming. So that's what your sarcomeres are designed for, the optimum zone of overlap. If you have a reduced zone of overlap, like right here, it's too stretched out already, those sarcomeres are not going to be able to contract that much because there's not that many cross bridges forming. Here, the zone of overlap is non-existent. The sarcomere is already stretched out too much. There is no zone of overlap. And then vice versa. Here, our myosin is already at the Z line. There's nowhere for it to get any shorter. It's already as short as it can be. It can't crunch down on itself, right? It's a rigid structure. So again, there's an optimal length that sarcomeres need to be. Let's talk about muscle twitch. This is when a single stimulus is going to stimulate a muscle fiber. It can vary due to muscle type, which muscle we're talking about, where the muscle is in the body, any external and internal conditions. Other factors are all going to affect twitch. You've all had that annoying eye twitch before. I know you have. That's what we're talking about. So you can measure the amount of tension that's brought on by a muscle fiber by doing something called a myogram. All those things hurt if you've ever had myograms done. It's basically looking at the amount of tension that's developed on a single muscle fiber. And it uses this process called fasciculation. Fasciculation is an involuntary muscle twitch under the skin. So it's usually looking at contracting a small group of muscles, a fascicle, right? A small group of muscle fibers controlled under one motor neuron. So let's take a look here at some specific muscles that undergo twitch. You have your eye muscle. That's that little eye muscle I was mentioning that can kind of go off. A high amount of tension, but in a very short amount of time. It's a small muscle. It doesn't take long to develop that same tension in it. Your gastrocnemius, your calf muscle, again, same thing. You can get a twitch there. It takes a little bit longer for you to get that maximum tension and for it to be relaxed. Same thing. The deep muscles in our calf, our soleus muscle, that undergoes twitches as well, fairly common. It takes a very long time for it to get to full tension and for it to relax. So a muscle twitch has several phases to it. There's a latent period. This is from the time that the stimulus comes to when tension starts to develop. And that's basically the time it takes for that calcium to be released. If you look back at this drawing, you see the stimulus was here at 0, but this latent period was about 2 milliseconds for every muscle group. Then you have the contraction phase. This is from the beginning of tension development. So after calcium was released at the end of that latent period, now our cross bridges are starting to form and that twitch is occurring. Then you get the relaxation phase. This is from peak tension to the end of the twitch. Calcium levels are falling, reuptake is occurring, cross bridges are detaching, myosin is reactivating, etc. Tension basically returns to resting levels during this time. And so here you see the breakdown stimulus, a couple milliseconds happen, buildup of calcium, cross bridges forming, you have tension building, etc. And then you get to maximum tension, and now calcium is beginning to be taken back into the terminal cisterna, and the cross bridges are detaching. You can pause the video here, go back and review. So let's talk about how tension is produced. Tension is produced by either how many muscle fibers are stimulated, or how much tension is produced by a single muscle fiber. So it's kind of quantity and quality. There are several levels of muscle tension. The stimulation frequency can affect how that tension develops in a single muscle fiber. And so there's 4 levels of how tension develops in a muscle fiber. You have something called TREP, you have wave summation, you have incomplete and complete tetanus. So let's talk about TREP. TREP is when a skeletal muscle fiber is immediately stimulated after relaxation. So it's basically beating that muscle fiber over the head with stimulus, basically getting that muscle to ultimate tension very, very quickly. Most skeletal muscles don't exhibit TREP. Some of them do. It's not as common. Again, being hit over and over and over with that stimulus, and notice each time getting eventually to maximum tension. Then you have wave summation. Wave summation is when a single or group of skeletal muscle fibers are hit with a stimulus before the relaxation phase has complete. So in TREP, here you're getting up, down, up, down, up, down. In wave summation, you're kind of getting this additive effect that's going to keep building and building and building and building and building the tension even past maximum tension. Incomplete tetanus is when a very rapid contraction cycle happens and there is some relaxation and you get almost to peak tension. So you're not quite getting all the way maxed out. And so that's why we call it incomplete. Kind of like wave summation, but we're not making it all the way up. Then you get complete tetanus, where there is a very high stimulus frequency and there's no relaxation phase at all. So no calcium ions are taken back into the sarcoplasmic reticulum. They're kept there, and the myosin heads just keep going and going and going. So this is peak tension being developed, continuous contraction. Doesn't usually occur in our normal functioning muscles, but it can. And this is very, very painful, as you can imagine, your joints or your muscles kind of locking up and reaching peak tension can be very painful. So let's talk about how our muscles develop peak tension. It's all about motor units and recruitment. So in other words, your motor neurons, as they come down from the nervous system, they branch off in several ways, right? And all of their axons branch off as well, the collaterals, and they go to different muscle fibers and muscle fascicles. So the same neuron can have an influence over a very large area of muscles. And that's what a motor unit is. So when that motor neuron is stimulated, doesn't matter where those axon sorry, where those axon terminals are, that muscle fibers, or those muscle fibers, if there's several of them, are going to be excited and they're going to contract. And so that's called a motor unit. A single motor unit has the motor neuron and all of the muscle fibers that it innervates. So the size of the motor unit is going to vary with control. Something like your external eye muscle, which has very precise control, only just has 4 to 6 muscle fibers with it. But a leg muscle, like that's just, flexing and extending, there's not very precise movements with it. There's there can be up to 2,000 muscle fibers with each nerve. So again, much bigger bang for your buck with those motor neurons. So here you see what I mean. This is like the triceps muscle, and all these different colors are just showing the different motor units, and they're kind of sprinkled throughout the muscle group. So in other words, when I'm flexing my bicep, it's maybe about 1/3 of the actual muscles muscle fascicles here that are actually contracting at any given time. They're kind of rotating. As I'm telling my arm to contract and to make this muscle, they're taking turns. And that's to reduce fatigue, reduce wave summation, reduce incomplete tetanus, right? So our motor units take turns being stimulated. And that's where we get this idea of recruitment. Recruitment is when the smaller motor units are activated first. Then the larger, more powerful motor units are activated. And so this is where there's more of a slow, smooth, and steady increase in muscle tension instead of just, oh, it's there already, right? There's also asynchronous motor unit summation. This is because the motor units are rotating to maintain that sustained contraction. So we call it asynchronous. They're not really synced up with each other. They do their own thing at their own time. So it's prohibiting that wave summation that we saw. So again, it's the tension in the tendon is relatively high, but the tension in the muscle fibers are not. And this introduces us to the idea of muscle tone. Muscle tone is the resting tension of a muscle. How many of us say, well, I want to go to the gym to work out, but I just want to get toned, right? Well, what you're actually saying is you want your muscles, even when you're resting, when you're sleeping, when you're watching TV, when you're riding in your car, you want your muscles to remain a little bit contracted so they feel firm, right? That's what being toned is. Your muscles have a little bit of tension in them, even at rest. So the resting tension is going to be higher in your toned muscles. So this ultimately is just a variable number of motor units that are being recruited over and over and over again. And this is regulated at the subconscious level. Yes, I can sit here and be like, oh, I want to tone my abs. I'm just going to tighten up my abs as I'm sitting here. But as soon as I forget to consciously do that, my stomach's going to be hanging over my pants in no time, right? So that's basically the difference between consciously saying, okay, I'm going to sit upright, I'm going to suck it in, or just naturally having that posture or that tone to your muscles. You can pause the video here and go back and answer these 3 questions. We're going to keep moving with the difference is in 2 main types of contractions. You have isotonic contractions. Isotonic contractions are about a muscle that shortens in length, but creates a high amount of tension. So the tension rises and the muscle length changes. So this is when you're lifting something. This is when you're flexing that bicep. You have concentric and eccentric contractions. A concentric contraction is when your muscle tension rises until it exceeds the load, the muscle shortens, the tension remains constant. This is flexing that elbow, holding a weight, curling the dumbbell, right? The speed of contraction is inversely related to the load. So in other words, you do it really fast, and that means your load is probably very light. If you do it very slow, it's probably something very, very heavy. Here you see that concentric isotonic contraction. Shortening, same amount of mass. Here you see it again, where we have our muscle tension increasing, we're getting peak tension, and the amount of load is relatively small. Here, our load is much higher. Same graph there. Eccentric contractions is when the load is more than the peak tension that's produced. So this is when our muscle actually lengthens. This is returning the dumbbell back to the flex position, right? Putting the weight back down. This could tear the muscle. It could break the tendon. It could, or your muscle could just recoil and go back to shape. So it, again, depends on how stretched and how overworked your muscle was at that point. It's a little stretched out. Now we also have isometric. So we were talking about isotonic contractions. That's when the muscle shortens and changes length. Isometric, distance or length, right, is staying the same. So the muscle length doesn't change. You can do this just by sitting here. I'm going to make my muscles, right? I'm tensing my muscles, but you don't actually see my arms flexing because I'm not moving them. The muscle length doesn't change. Tension never exceeds load in this case. The contracting muscle bulges, but it doesn't bulge as much as during an isotonic contraction. And this would be like postural muscle contractions. I'm going to flex my abs. I'm going to flex my arms even though I'm just sitting here and you can't really tell that I'm doing it. Right, there they are. So you can pause the video, go back and answer these 3 questions. Moving right along, let's talk about how energy is produced in muscles. There are a few sources of energy in muscles. One is free ATP. This is very minimal, small twitches, etc. are supported by free ATP. Creatine phosphate is one of the first energy sources in our muscle cells. It's usually abbreviated CP. The creatine is assembled from amino acids, so it's a protein-based compound. It's basically going to facilitate the regeneration of ATP. By doing that, creatine phosphate is basically donating the inorganic phosphate from it to the ADP molecule, creating ATP plus a creatine. Glycolysis, or anaerobic respiration, which is just producing 2 ATP molecules and some lactic acid, and then aerobic metabolism, producing a lot of ATP, but it requires a little bit more time, and of course it requires oxygen, which requires you to be breathing efficiently and efficiently getting that oxygen to your muscles. So if your body's not trained or not used to delivering oxygen to all of your tissues, then you're not going to be doing too much aerobic respiration. It's going to be mostly glycolysis. You're going to build up lactic acid, you're going to feel the burn, and you're probably going to get fatigued very quickly. That was an aside. All right, back to the PowerPoint. Most energy in the muscles is stored in the form of glycogen. Glycogen is basically compacted glucose, right? If you remember, when we bring in food to our body, it's broken down through the process of hydrolysis, digestion, and all of that glucose floats around our blood, gets taken up into our cells. And if our cells aren't using that glucose, our cells are going to pack it up in the form of glycogen stored in the liver and skeletal muscles. Well, now that we're finally using all of this glycogen, maybe we just started an exercise routine or started walking in class or whatever, your skeletal muscles specifically are going to start breaking down this glycogen. So the process is glycolysis. So glycolysis does not require oxygen. The glycogen is broken down in something called glycogenolysis. It's just a metabolic term we'll learn in chapter 15, I think, next semester. But either way, glycolysis is the first thing that occurs to a glucose molecule in order to get it to create ATP. A net 2 ATP are produced, and 2 pyruvates, or 2 pyruvic acids, are produced in this process also. Aerobic metabolism gets the majority of the ATP demands for a muscle cell. This happens in the mitochondria. This is mostly due to the electron transport chain, the citric acid cycle, or tricarboxylic acid cycle. It produces about 17 ATP for each pyruvate. So if each glucose gives you 2 pyruvate, you're getting about 35 ATP from 1 glucose molecule. Which, just to give you a frame of reference, I Googled it. I don't know how good my source was. It wasn't Wikipedia. It was a good source, but I didn't cross-check it. But I read that blinking your eye, just one eye, not winking, but blinking, just one eye, is about 20,000 ATP. Crazy. So every time you blink, that's 40,000 ATP. That sounds crazy to me. But I don't know. It is just one little molecule. So I would be interested if you would challenge me on that, please, because I want to know the real answer. It sounds kind of off, but that was the only thing I found, and I saw it on a few different sources. Side note, back on topic, sorry. Here's the whole process. This is inside of our cell, this little yellow box. Glycolysis happens in the sarcoplasm. Glucose is broken down to pyruvate, gives us ATP. Pyruvate goes into the mitochondria, goes into the citric acid cycle, which releases carbon dioxide. It also produces these little protein carrier molecules, NADH and FADH2, which take hydrogen ions over to the electron transport chain, use that potential energy to create water and ATP. If I went over that too fast, that's okay. There's a whole chapter next semester dedicated to all of that. Take a look here. You'll see exactly what I was just talking about, the sources of energy, I should say, in a muscle cell and how it is used. So let's talk about how energy is stored. We just talked about how energy is used. Energy is stored in the form of ATP, creatine phosphate, and glycogen. Glycogen is a very small percentage of the weight of a muscle, but it enables our muscles to extend their activity for a much longer period of time. Even at rest, the demand for ATP is relatively low. Fatty acids and glucose are absorbed into the bloodstream, and they're metabolized for the creation of ATP. We also use that to build up our creatine phosphate reserves and glycogen reserves. So this is a muscle at rest. All this stuff, our body's like, sure, come on in. We're not doing anything anyway. We may as well all these fatty acids and glucose into the muscle cell. We'll stock up on ATP and creatine phosphate. Why not? Then as your moderate activity levels increase, your demand for ATP increases. So ATP production is going to basically meet the demand. And pyruvate is now beginning to not be stored, but it is beginning to actually be used and metabolized. So you're going to start using oxygen more. And this is when you start to increase your respiration rate, right? Because as your metabolism goes up, your demand for oxygen goes up, your respiration rate needs to go up so you can bring more oxygen in. So you won't feel any fatigue until your glycogen, lipid, and amino acid reserves are exhausted, which will basically be a very long time. Your body can do it. You just have to tell your mind to get over it. So this is moderate activity levels. Fatty acids are still coming in. Oxygen still coming in. Glucose is not coming in anymore because we're taking the glycogen that we already have, using it to make glucose, using that glucose to break down into pyruvate to make ATP. So at peak activity levels, now we have huge ATP demands. Mitochondria maximally producing ATP as best we can, but it's only meeting about a third of the ATP demand. So the majority of the ATP demand at a high level of activity is coming from glycolysis, which is anaerobic. What's happening with the anaerobic metabolism is all this pyruvate is building up. It's kind of like a traffic jam. It can't enter the citric acid cycle because there's not enough oxygen there. So all this pyruvate builds up and it gets transferred, or what's the word I'm looking for? It's late at night. It gets changed into lactic acid. And so this lactic acid builds up, and that is what makes you feel the burn, right? It's going to lower the overall pH of your muscle cells. It can damage them as well. It can alter enzymes, can be very dangerous. So here's what we got at high energy levels or at high activity levels. Notice nothing's coming in. Our muscle cells are saying, whoa, we're contracting a lot here. I can't handle anything else. I just got to do what I got to do. I'm contracting. Just leave me alone, right? So glycogen's breaking down to glucose, pyruvate, and notice there's no citric acid cycle. There's no electron transport chain. There's nothing going on with lipid metabolism anymore. It's strictly a buildup of pyruvate, which is creating lactic acid, which is being converted into lactate, leaving its hydrogen ion around. Hydrogen ions, that's an acid. So the pH of our muscle is going down, down, down. You can pause the video here, go back and answer these 3 questions. Let's talk about fatigue. That's the next step. So our muscles are just going to keep working harder and harder and harder, and lactic acid is going to continue to build up, build up, build up, and that is when we reach the point of fatigue, when a muscle can no longer perform the activity that we want. And the major factor here is that decrease in pH. When there's a decline in the pH, when the pH levels go down, it's going to decrease the ability for calcium to bind, and it's going to alter all of our enzyme activity. So the ATPase can't work on the myosin head, it can't be detached, it can't be recharged, etc. So under other conditions of insufficient oxygen, glycolysis can quickly produce ATP, as I said, in that anaerobic respiration, but we're going to have that lowering pH effect. Lactic acid is going to build up, faster ATP production than an aerobic metabolism until glycogen reserves have been depleted, but it's much less efficient. Only 4 to 6% of energy captured from the conversion of glucose to pyruvate. So it's not nearly as efficient as the citric acid cycle and the electron transport chain. Overall, having insufficient oxygen in the body and using anaerobic metabolism is going to increase the body's temperature because we're trying to sweep out all of that oxygen or out of all of that acid out of our muscles, and that's going to trigger sweating, right? Our overall homeostatic mechanisms. In insufficient oxygen, glycogen is being converted to glucose. Glucose is being converted into pyruvate. Pyruvate's being converted into lactate. We're not dealing with the mitochondria when we don't have oxygen. Under conditions where oxygen is available, like during a recovery period, intracellular conditions will return to normal. It can take hours or days. Let me tell you, I'm training for a marathon right now. I'm about to go for a 22-mile run in a day and a half. I'm resting, I'm carb loading, and I know that for about 3 days after I do that run, I'm not going to be in good shape. My legs are going to hurt. I'm going to be stiff. It takes a lot out of you to push your body to that level. And it's not just me not being in as good a shape as I probably should be to run a marathon. It's also your body needing that time to recover and reestablish the oxygen in your blood and the your muscle cells to repair themselves under those acidic conditions from the lactic acid. So there is a recovery period that we all know if you've ever worked out or pushed your body's limits a little bit. And that's when that oxygen can then be available in abundance. ATP production returns to mostly aerobic metabolism, which is much more efficient than glycolysis. And that recovery period is also when that lactate is converted back into pyruvate, and that pyruvate can go back into the muscle cells and then be used for aerobic metabolism, or to be converted back into glucose, to be converted into glycogen, to be stored for later. So here you go, the whole process. Now that we have oxygen, yay, all the lactate that's been floating around or being stored in the liver can come back, be converted back into pyruvate, go back into glucose, be stored as glycogen so we can use it for later. Or we can use it right now for energy, release the carbon dioxide, create the ATP, the water, etc. That lactate is recycled, as you just saw. It's a cycle called the Cori cycle. It involves the liver. The lactate that's produced during strenuous activity goes to the liver from wherever skeletal muscle is being produced. The liver reabsorbs it and basically recycles it back into glucose. It actually recycles it into pyruvate. And then during peak activity, the lactate that's produced goes into the blood. The liver begins converting that lactate back into pyruvate, and pyruvate can then be used for ATP production. During that recovery period, the liver is converting the lactate into pyruvate and then into glucose. So the majority of the pyruvate is converted into glucose. So some of it is broken down and used for energy. And the glucose can then be released into the blood and then go back out to wherever it needs to be absorbed and even be stored back in the form of glycogen. So from producing ATP in the muscle cells and liver, the oxygen demands are relatively high. That's what we call an oxygen debt. So this is an excess post-exercise oxygen consumption, or EPOC, basically the amount of oxygen needed to return to pre-exertion conditions. And this is what I was referring to when I was talking about my marathon training. I know that it's going to take a few days after I get done running for my body to reestablish the oxygen reserves that are throughout all my muscle cells. So here's lactate production and recycling in the Cori cycle. During peak activity, you have all this lactate being put into liver. That's going to be converted into pyruvate. Some of it's going to be used by liver cells to create ATP. Most of it's going to be used to go back to glucose. The glucose can then go into the blood, go back to the muscle cells, be converted into glycogen. Glycogen can go to glucose, and so on and so forth. So the Cori cycle is basically the creation and recycling of lactate. All right, you can pause the video here, go back and answer these 3 questions. Let's talk about different types of muscle fibers. You probably know, and again, I'm just talking about running because I'm kind of a runner. You probably know, if you've ever ran before, if you've ever participated in sports before, some people are just naturally faster than others. Some people have better endurance than others. I can run for days, but I'm not very fast. Other people are very fast, but they don't have that much endurance. It has a little bit to do with genetics, and the genetics piece is related to the types of skeletal muscle fibers that they have. You have fast fibers, slow fibers, and intermediate fibers, and they're pretty much as they're described. So let's take a look at them. Fast fibers reach their maximum tension very, very quickly. They have a very large diameter, They're very densely packed with myofibrils. They have lots of glycogen in them, very few mitochondria. So if they have few mitochondria, how are they going to do aerobic respiration, right? They're not very good at aerobic respiration because they don't have a whole lot of mitochondria, but they produce very powerful contractions, but they fatigue quickly, again, because the ATP can't be produced very, very quickly. So you have fast fibers usually in your small muscles, like the muscles of your eyes, which can move very, very quickly, but you're not going to be going like this with your eyes, moving those eye muscles very quickly and using up your ATP reserves in your eye muscles. That's not a very common thing. You also have slow fibers. Slow fibers are half the diameter of fast fibers. They take 3 times as long to contract, hence slow fibers. They can maintain sustained contractions. They're usually used for aerobic metabolism. They have increased oxygen networks. They have something called myoglobin, which is a pigment that contains extra oxygen, and that makes them appear a little bit darker and red. This is red meat, so to speak. It's kind of creepy to think about on a human, but it's dark meat. There you see the slow fibers in a cross section. Then you have intermediate fibers. Intermediate fibers more resemble fast fibers, but they don't have as much myoglobin. So they're relatively pale, and they have more capillaries. They're more fatigue resistant than fast fibers. But there you can see fast and slow fibers. And here's just a quick chart of a comparison between them. Let's talk about how most muscles have a mix of these fibers. So the percentages of each type are obviously going to vary according to the muscle. Your back muscles, your calf muscles, mostly slow fibers, right? Your back is extended all day long. If you're sitting upright, if you're standing, your back muscles are contracting to keep your back extended, to keep your torso extended. So they have to be slow muscles in order for them not to fatigue. Hand and eye, because the movements are precise and because usually you're not doing things with your hands over and over and over, your eyes over and over and over again, they're fairly, they will fatigue fairly quickly. Go to my lecture. I bet you're maybe taking notes right now and maybe you've had to pause the video a few times because your hand is hurting you. Thank your slow fibers, thank your fast fibers for that, because you don't have as many slow fibers in your hand, so it fatigues quicker. According to genetics, fast and slow is inherited, as I was saying before, and physical training can help modify some of your fibers. So again, as I was saying, I'm more of a distance runner, I'm not a sprinter, but if I wanted to be a sprinter, I could practice and I could still gain speed. Would I be as fast as someone who has more of those fast fibers? Probably not, but athletic training always helps, just like, with anything. Diet and exercise, etc., etc. You can pause the video here, go back and answer these 3 questions. Moving on, let's talk about some clinical cases with muscle or muscular system. Hypertrophy, this is just the growing of our muscles. This is typically due to an increase in myofilaments, an increase in myofibril size, and an increase in mitochondria. More glycogen, more enzymes, etc. It's not because you're getting more muscle fibers. You don't actually grow muscle fibers. Your muscle fibers are pretty much all you're going to have for the rest of your life. So when your muscles are growing, when you're exercising, when you're getting bulked up or whatever, it's really just all of these things that's happening. Myofibrils, myofibers, filaments, etc., getting bigger, not the actual muscle fibers. And you can. He has the same number of muscles as I do. I just don't look like that. All right, atrophy is the opposite. Atrophy is a decrease in muscle size, muscle tone, and power/strength/tension, right? This could be because of paralysis, damage to the nervous system, maybe having a body part that has been immobilized by a cast or something like that over a long period of time. I'll tell you this, I broke my distal radius last summer. And I was in a full arm cast and I couldn't move my hand, because the cast was wrapped around my hand. And you don't think about, look at my hand, right? It's not flat, and your hand probably isn't flat either. But when I got that cast off, my hand was flat. It was the weirdest thing. It literally, I don't have anything flat right here. Oh, here's my Kindle. It was like flat, completely. There was no muscle tone. There was no meat on it at all. I had to do lots of hand exercises, squeeze those little squeezy balls to get my hand strength back up because I had no muscle. I think I was in the cast for 8 weeks, something. It was a long time. Anyways, atrophy can be reversible as long as your motor neurons are still there, still able to fire. But if you're paralyzed, obviously, you have other issues that may impact the reversal of atrophy. There's atrophy. Not a lot of muscle on that hand. There's also muscular dystrophy. Muscular dystrophy is an inherited disease. It's kind of a category of diseases where the muscles will weaken and deteriorate over time. There's Duchenne or Becker muscular dystrophy. This is the most common form. It appears in childhood and affects mostly males. 1 in 3,500 to 5,000 males are born with it. It is a sex-linked trait, which means it's carried on the X chromosome, which is why males are more likely to get it than females. It's very progressive. So the babies are born, they grow up a little bit, a few years later, they're walking, talking, moving just fine, and then all of a sudden you notice that they're having trouble walking, or they're stumbling, or they have reverted back to crawling. Most individuals that have Duchenne muscular dystrophy end up dying from respiratory issues because, again, the respiratory muscles can't contract as they need to. There's also polio. Polio is a virus that attacks the motor neurons of your brain and spinal cord, and that is what causes paralysis and loss of voluntary movement. Very sad. Tetanus is a toxin from bacteria, which suppresses the inhibiting of motor neuron activity. So, otherwise, your motor neurons are firing overdrive, and your muscles are having very powerful, sustained contractions, producing tetanus. 40 to 60% mortality in tetanus, right? Imagine your respiratory muscles being paralyzed. You wouldn't be able to exhale, right? Or you wouldn't be able to inhale. And botulism is another disorder caused by bacteria. This is going to block acetylcholine receptors. So this is going to basically paralyze your skeletal muscles. Myasthenia gravis. This is a loss of acetylcholine receptors at the neuromuscular junction. It's a progressive muscular disease. Rigor mortis is a clinical condition, although you're probably familiar with that already. This is when a generalized muscle contraction occurs a couple hours after death. Think about the role of ATP in muscle contraction. You need ATP to remove the myosin head from the cross bridge. So once your body stops metabolizing, once your ATP reserves are used up, those myosin heads can still be attached to the actin, which means your muscle is contracted, but there's no ATP to release it if your body's not metabolizing anymore. So that's what rigor mortis is. It typically ends as the muscular tissue decomposes a couple days after death. But in forensics, it's one of the ways that you can determine time of death by looking at how contracted those muscles are. And this is the last slide. You can go back and answer these 3 questions. Let me know what questions you have about this chapter. I did go through pretty quickly, but I tried to break this chapter up because I kind of felt that all of the excitation, contraction, coupling, sarcomere stuff, and then all of the other stuff that goes with the muscular system could kind of be split up into 2 little PowerPoints for you. And so anyways, let me know if you have questions. I'll see you in lab, and have a great day. Bye.